All right, so for this lecture, I want to just sort of overview some of the immune system here and what to expect, and start getting into some of the details about how this works. So just right off the bat, I just want to point out that a lot of times we think of immunology in the context of microbes or pathogens. However, as depicted here on the background, you're seeing a response from T cells, which are important cells of the immune system, and they are attacking a cancer tumor cell right here. And so it's not just microbes. Immune system plays a role in clearing our body of cancers and just also tolerating our own cells, which are within our body. So I think this story makes the most sense when you actually get to see an example of what infection looks like. So I want to start off with a basic example of just a generic infection. What we have here is a layer of the skin. We have our squamous epithelium right here, stratified squamous epithelium. Underneath that, this basement membrane, which separates the lower underlying tissues from the top. And we have here some of the blood vessels which run through this. And so over here, this is just a generic cell for now in this example, but we can typically think of sentinel cells which hang out in these tissues, one of them being a macrophage. And so in this example, this macrophage is in the tissue. When suddenly there's a break or exposure, this allows microbes then to come in through the tissue. And this microbe, doesn't matter what it is for now, will take up residence and begin doing the things it does to replicate and cause disease. The macrophage will move towards it and make an attempt to engulf it. And when it does this, it has a number of sensors which can broadly detect what this thing is. And once it does that, it will send out these chemical signals called cytokines to other members of the immune system. This acts on the blood vessels. It creates a swelling of that blood vessel, allowing more cells to come in, soluble proteins in the blood to come into the tissue where the infection is occurring. And so this is a really important initial step in starting the immune response. So meanwhile, more microbes are making their way into this cut while this immune response is mounting, until finally the neutrophils, in this example, they are these red spiky cells. The neutrophils arrive and they really begin the first wave of attack on the microbes. They will try to ingest these microbes. They will try to dump toxic chemicals on them, reactive oxygen species, and this is their attempt to just non-specifically destroy whatever that macrophage called it into the tissues for. While this is occurring, there is another sort of brains of the operation type cell, which we call a dendritic cell. And this cell is really meant to transition from this innate immune response into what we call the adaptive immune response. And this is eventually what will clear the infection completely. But this cell will gobble up some of that microbe and it'll take it off to a regional lymph node. And there it begins searching for lymphocytes in that lymph node, which would recognize the pathogen and are specific to that pathogen. It takes a little bit of time for this to occur, however. So during that time, that infection is starting to wane in the favor of the microbe in that there's only so much that the innate immune system can do, and it may keep it at bay long enough so it doesn't get bad. But in this example, the microbe's really starting to win out. And fortunately for the host, just in time, the dendritic cell, after a few days, typically a week, 5 to 7 days, finally rallies enough of these lymphocytes and gets them going and alerts them to the presence of this microbe. And so these arrive over here, and they're depicted by these different colored spheres. And we can say that these are CD8 T cells, these are CD4 T cells, and this is a B cell, secreting these soluble antibodies depicted by these yellow stars. And I'll go into more detail on what this all means in the next video, but these lymphocytes really take care of this infection. They're the ones that eventually will clear this, and they will typically develop some sort of lasting memory to whatever this microbe was. Once they do that, they all move out of there, and the neutrophils that are left will die off. And so that's just a basic overview of what happens during a canonical, just generic infection. Some important terms that you're going to need to be familiar with so that you're not scratching your head when you hear them, like perhaps you did just now. We have terms like antigens. So these are any substance which a lymphocyte will recognize through its receptor or through the antibody. Cytokines, which are these secreted soluble proteins which direct immune responses. There are ways of they're the ways that cells can communicate between cells and send these vast messages throughout the tissues. And generally what they do is cause some sort of differentiation, or they'll cause some sort of development for cells that can respond to them. We also have chemokines, which are a subset of cytokines. These are similar small proteins, and they tend to create a chemical gradient along which immune cells can follow. And as that gradient gets stronger, the immune cell knows that it's moving towards the source of that chemokine. Antibodies, I've mentioned those. Those are probably something most people are broadly familiar with, but due to like vaccinations. But these are soluble proteins which B cells and plasma cells really. The B cell will grow into a plasma cell. Again, I'll mention that in the next video, but it starts secreting these antibodies, which really play roles in neutralizing pathogens, so it stops their function. They will coat pathogens, it's called opsonization, and they will coat them so that the other immune cells can see that very easily and recognize it and destroy it. And they also do this thing where they activate a system called complement. It's a fairly complex or involved cascade, so I'm not going to go into that here, but it's an important innate defense. We also have antigen presenting cells, and so these are any cells which can present peptides that they've ingested or that they have been infected with to their surface and allow other cells to see what they have inside of them. Host peptides get presented all the time, but the immune system is primed in such a way that it doesn't respond to those. So ideally, you're only seeing peptides and responding to peptides, rather, that are not of host origin. We call the dendritic cells, the one that ran off to that lymph node, the macrophage, the initial cell which was there, and then the B cells, which came in to produce antibodies. We call these professional antigen presenting cells for reasons I won't go into in this video, but we'll come back to it eventually. So just some more basics of the immune system. There's a lot of ways that this can be broken down and presented, and so I'm going to attempt some of that here. This is just an overview of what the immune system meant to do. It's this collective and coordinated response to pathogens, foreign substances, and even self-antigens, in this case, maybe cancer. We don't think of it as our own, but it really does arise from cells that were normal at one point, and they were your own cells. Additionally, though, the immune system plays a role in tolerating other cells which are not cancer. Just any cell in your body, you have to be able to recognize it as your own. And so that's what another function of the immune system is. It can be broadly characterized here as either active or passive, meaning you develop the immune response and effector cells on your own, or it could be passive in that they're transferred to you in some way. And this happens either naturally or artificially for both cases. So an active natural immune response would just be a direct native exposure to some virus or bacterium, and you develop antibodies and effector cells over time. We can artificially stimulate that through vaccination. So we're purposely giving a weakened portion of what that microbe looks like, and then your body generates an active response on its own from there. And then we have, like I've mentioned, the passive immunity. So an example here would be the pregnant mother gives birth, and she's now breastfeeding the baby, and her antibodies, which she made, are being passed on to the baby. And so she's conferring immunity to the baby passively, and that's through a natural route. She produced them. Whereas artificial might be something like a medication that you take, which is a neutralizing antibody. That antibody isn't, even though it's made in the lab, it's made in the lab by cells. And so it's not like we engineered it. It's the native antibody. It's just made in a lab somewhere and then purified and packaged. That way you can inject it. And so that's an example of passive artificial. The immune system protects in two main ways: through humoral, meaning blood components, and also through cellular components. So some of the blood components would be those antibodies or the complement system, and then the cellular components are just like they sound, they're cellular, they're cells. There's two major lineages, which I'm going to go in the next video about, but these are just broadly, they're myeloid and lymphoid lineages, and they're broken down sort of into two branches. The myeloid cells tend to group with innate responses, whereas the lymphoid cells tend to be more of the adaptive response. And so just to highlight some of these general features about the differences, what you're seeing is a picture of some of the cell types, and these are of the innate immune system with a crossover here of these cells which straddle innate and adaptive. I'm not going to go into the cell types now. I just want to give you an appreciation for how many there are. Really the function of this system is to be this first line defense. It's always present. It's present for birth. It doesn't require any prior exposure to learn it. And it serves a number of protective layers in that there's anatomic barriers like your skin, which physically blocks microbes from getting in. There are chemical barriers like low pHs of the stomach. We have really salty and low pH skin. We have chemicals that are secreted in like tears, and antimicrobial peptides that are present, antimicrobial enzymes. These are all ways that the immune system always has this present chemical barrier ready to try to kill anything that invades. Innate immunity is dominated by the phagocytes, which I'll go into again at another time, but these are cells that engulf pathogens. And then also that complement system, which is always present, serving a role in identifying and tagging microbes. And so this system, since it is always present, it activates rapidly. And it does this based on, not on specificity of a microbe, but rather more of these conserved molecular patterns that the microbe might have. So if you think back to the Tree of Life video that I was talking about, what I mean by conserved is we know that all living organisms are related to each other through this lineage of descent with modification. And so a lot of times what will happen is we'll find structures or sugars or proteins that remain throughout a number of different bacterial species and even genera. And so we call that conserved. It's some sort of thing, whether it be a protein or some sort of adaptive trait of that organism, which works really well. And therefore it's sort of, it can show up in other species as well. And it continues to be useful in some adaptive sense. And so I've said these things here, dominated by phagocytes. The other major part to this is the immune system, the innate immune system, which has this dendritic cell. This is the adapter for the adaptive response. It sort of straddles that line between between recognizing that there is an innate immune response going on, and then promoting the involvement of the adaptive immune arm. Here we have the adaptive immune cells, and these are of the lymphoid progenitor. And so the other ones were myeloid. These ones are lymphoid derived. And the major characteristics here then are that this response takes time. It's a delayed response because the cells have to be sought out by that dendritic cell. They have to be activated, and then they have to grow and proliferate and then make their way back to that tissue where there's the infection. And so that takes some time. That's really the main purpose of the innate immune system, which is to stall until this system can get up and running. Repeated exposure to the same pathogen activates the system even faster. So if you are exposed to, let's see, I don't know, you pick your favorite bacterium or virus. Typically, not always, but typically, because of some genetic reasons, that's why I say that, they can escape these immune responses. But in a perfect world, if you encounter some sort of microbe, the second time that you encounter it, a week or two or a month later, the adaptive immune response doesn't have to go through learning what that microbe is anymore. It already has seen it before, and so it just has to call into action these immune cells which have already seen this at one point. It's very good at discriminating between self and non-self, unlike the innate immune system. It's highly specific to the antigen, which it knows, and it has both cellular and humoral components. And what I have here, so this is this system is really dominated by these lymphocytes, which are a subset of all cells in the immune system called leukocytes. And just a couple more, 2 slides here of just definitions before I wrap up. I'll leave this here for a moment so you can read it, and you can pause if necessary. I'm not going to read it word for word. I just want to touch on these highlighted parts. Epitopes are specific amino acid sequences or structural sequences or motifs that are physically recognized by lymphocytes or antibodies. So you can have an antigen which has multiple epitopes that are recognized by different antibodies. Each epitope is unique though. It's sort of a signature of that microbe. Memory, I've touched on. Memory is just the ability to have these leukocytes, specifically the lymphocytes, stored in some sort of capacity where they can be recalled rapidly and you don't have to go through that learning process again. And over time, this does wane, but it can be boosted by re-exposure, which is in part, well, really the main reason why we do booster shots, for example, is to re-educate the immune system so we keep that population of antibodies up really high. Let's see, intolerance. I'm not going to go into details here. You can read those if you like, but really tolerance is just to get the point across that the immune system is trained. It's a deliberately trained system in which it has to learn what self is, and that happens during development. And I think it's really an interesting concept. Even though that these cells are of our own body, they will attack our own body. And we see that in things like autoimmune disease. And this is just one last slide here talking about the way in which those lymphocytes are able to recognize antigen. And again, there's a lot of text here. You can read it. I put it really just for completeness. The thing to take away from here is that lymphocytes don't typically, at least there's 2 different ways lymphocytes will recognize antigen. And with the B cells, they have the antibody complex, which can recognize fully folded native proteins. However, the T cells can't do that, and they have to recognize the microbe in the context of something of their own, which is presenting that antigen. And that thing is this major histocompatibility complex, and it's a protein which will house a short portion of the pathogen's peptide, and it'll hold it out to the T cell, and it'll say, do you recognize this? And the T cell will look at it, and if it does, along with the number of other signals that are going on, it will decide to either turn it off because it wasn't a pathogen, or it will decide to activate and proliferate and then go find that pathogen. But the MHC, major histocompatibility complex, I just wanted to introduce it. It's a really important concept and definitely worth knowing. So in the next video, I will cover some of the cells, the most important cells which are involved, and some of this hopefully should make continue to make more sense. So thank you for tuning in.